Okay, for this video segment, we're going to carry on after replication has taken place. And so if we go back to replication, basically, from this slide, you see the DNA unzip. There's enzymes involved there. Keep in mind you are responsible for those enzymes. Like here we've got helicase that does the unzipping of the DNA. It's breaking the hydrogen bonds that are right here, so it goes in and breaks those, separates, and then the other enzyme would be polymerase, and that polymerase is putting all those new A's, T's, C's, and G's. I'll just put the letters there, A's, T's, C's, and G's, and it just basically makes two strands. That's the idea of replication. Now what we want to do is the actual process of, gene expression. And what do we mean by gene expression? Those are all of the traits that you have coded in your DNA that you received from your parents. Remember, you get half of your DNA from mom, half of your DNA from dad, and those strands of DNA are loaded with genes or traits. Think about eye color, think about hair color, the shape of your nose, the length of your fingers, skin color, all of those things. That's the idea of gene expression. And so what we want to do, this picture, where there's a crazy amount of stuff going on, we're going to try to make sense of it. And so for transcription, that's this part of the picture. That's right here. And we're going to try to make sense of this part of it. And then when we get to translation, so here, there's transcription. Out here, there's translation. And so there's two different things going on, but the first of which is transcription. So that's what we're going to take a look at in this segment. So we're using DNA to make proteins, and location is important. So replication, these are things you'll want to know. Replication occurs in the nucleus, and transcription occurs in the nucleus. We haven't got to translation yet, but when we get to translation, location-wise, that takes place in the cytoplasm. And so to differentiate here, replication, is making two new copies of the DNA. And then what we're going to take a look at right now is transcription, where we want just a segment of the DNA copied, and then that segment can leave the nucleus. It goes out into the cytoplasm, and then it gets translated. And when we say translated, think of a code. When you were little kids, maybe you wrote codes, or think military, they use codes. Well, when you're sending codes, you have to be able to translate it. Well, those A's, T's, C's, and G's in our DNA are kind of like a code, and we have to translate those and turn them into our ultimate goal, which is that protein. We translate them into the protein. That's the actual gene expression part of it. So we see gene expression going from DNA to proteins, and that's our goal. So we have a new player in the game, all very similar to DNA. We see RNA, and again, we don't see it spelled out very often. DNA, remember, was deoxyribonucleic acid. RNA is just ribonucleic acid, so it's a slightly different molecule. They are not the same. And differences, DNA comes in two strands. RNA only comes in one strand, where it says half a strand of DNA. RNA is single-stranded. Not all the bases are the same. We have adenine, guanine, and cytosine, but it does not have T, no thiamine. There's no thiamine in RNA. And so that's a nice thing. Right away when you're looking at the strands, if you see a T, it has to be DNA. If you see a U, which is uracil, there's a different base. Whenever you see that U, you know it has to be RNA. So it's a really simple, easy way to tell the difference. Ribose is the sugar in RNA. And there we go, the last picture, there is that base, uracil. And so we'll see U's now instead of A's. So at the beginning, the U's will cause some confusion, but you'll hopefully get the hang of it. And if we go back to Chargaff's rules, RNA, remember, no thymine, so it's A to U. You know, so things are basically the same, but we notice right here, DNA has thymine, RNA has uracil. So we will see that difference. So be on the lookout for that, because you will see that quite a bit now. I would say 3 flavors. RNA comes in 3 flavors. It has 3 different jobs to do. So the 3 different varieties, and differentiated right here. M is messenger, and that carries the code copied from the nucleus, and that carries it out into the cytoplasm. We don't ever want to take the DNA out of the nucleus, but we can make a copy, and that messenger RNA is the copied code. Our RNA, ribosomal RNA, we talked about organelles a while back, and we talked about ribosomes are the places for making proteins. Carries a code for making ribosomes, and that equals and we saw that with our cells unit, that ribosomes are where the proteins are made. So our RNA is actually in charge of making the ribosome, and that's where the proteins are made. And then transfer RNA is kind of a helper molecule. It's carrying amino acids found in the cell to make the growing protein chain. And so it's transferring amino acids to the strand of DNA. So we will see all 3 of these. And again, the difference, we will see the M, the R, and the T for the 3 different varieties of RNA. So here's the process. So process by which the DNA is read and a messenger RNA copy is created. And so messenger RNA DNA is too important. It never leaves the nucleus. There we go. Messenger RNA, it's exactly that. It's the messenger. It carries the code out of the nucleus. And the idea is if something would happen to the messenger RNA, you can always go back to the nucleus where the DNA is located and make a new copy. So if you copy a code on a piece of paper and that piece of paper gets tossed in the wash and it gets wrecked, well, you can go back to the original code and make a new copy. And that's kind of the idea with the messenger RNA. And so then what happens is transcription complex, that's all you and I are going to call it. And then you and I, similar to replication, there's 2 main enzymes. There's helicase, which does the unzipping. And RNA goes along and adds the new bases. And so these 2 are found in just what we're going to call the transcription complex. There's more enzymes involved in that, but those are the only 2 that you and I are going to worry about. And so here we see the unzipping. We see the unzipping of the DNA because there is a segment that needs to get coded for. And so it starts to open up. And then as transcription continues, so nucleotides pair with 1 strand of DNA. And we see that red backbone we see right here. There's that messenger RNA, and I'm going to try to see if I can do some erasing. Always seems to be a treat when I try to do that. Got it all this time. So there's that strand starting to combine, and there we see that RNA polymerase bonds the nucleotides together. It's adding new bases, and then the DNA helix zips up again. So on one end, it's being unzipped, it's unzipping, and This isn't a very good term, but being rezipped. It's being rezipped. So as that transcription complex moves along the DNA, it's being unzipped on the front end, rezipped on the back end, and this growing strand of messenger RNA is what's being created. And there we go. Again, we see that RNA polymerase is moving along the DNA. Once we get to that end spot, so now here, all of these, those are that was the transcription complex is what I was trying to write. So those are the various enzymes. Again, remember, we're interested in the helicase and the polymerase. Those start to break up. And now this is the ultimate end goal of transcription. There is our messenger, I'm going to put a little M in there, messenger RNA copy. That's now free to leave the nucleus. It can go out into the cell. Eventually we're going to see that gets translated. And back here, our original strand of DNA is left in the nucleus. And so if we ever need to make a copy again, we just go back to the nucleus and transcribe another copy. And so now here we've got this messenger strand, A's, U's, C's, and G's. Remember, on RNA, there are no T's, it's U's instead. And so here we see this is just another view of the same thing. And so here we go. And so here now, RNA polymerase. We see that enzyme. This one isn't showing, remember, helicase as well. It unzips the DNA, so remember that DNA would be getting unzipped. And now that polymerase is moving down the strand of DNA. And now here it's showing it with the blue. There's our messenger RNA. That growing messenger RNA strand. And so now it keeps moving. Again, it's keeping moving. And here's that growing strand. And eventually it gets to a stop site. And now here, there's our messenger RNA strand that's free to leave. The DNA zips back up, and that transcription complex starts to break up. And it'll go get used again for another enzyme somewhere else. Not enzyme, sorry, protein somewhere else. And so if we look over here, if half the strand of DNA, let's go this way with it. If half the strand of DNA here, if we've got that GCT, there's our DNA. Remember, it's got the T's. DNA has T's. Well, G's bond to C's. C's bond to G's. A's would still show up in RNA, but here's where we see the difference. We now have here's an A. A would normally bond to T, but we don't have T's in RNA. That's where that U shows up. So A bonds to U, C bonds to G, A bonds to U, G to C. A would still show up. It has adenine. And so the only difference is we see U's no T's in RNA. And so there we've got just a different layout to the strands. And then this last one, this video clip doesn't work the best on the iPad, so this one you'll have to show from the laptop, and then that'll show the idea of transcription.